So I'm so pleased to see so many people here on Cinco de Mayo. Yay! Yeah, we're all in the crowd. Oh.
So you can take the mic.
Which is the mic here? Oh yeah.
So you can just remove it.
Yeah, I don't. Hey, yeah, I have to speak into the mic and it's on. And thanks for coming, everybody, to hear Rachel speak about her work at San Francisco State. It's been really wonderful having Rachel in the lab. She came here during the pandemic.
That's the camera.
Oh, hi, camera. So she came here during the pandemic, and of course, that was stressful because it wasn't an easy time to be at San Francisco State University when the lab was closed, right? So pretty difficult times. She started exactly in the fall of 2020, so you can imagine what was going on. The lab was basically closed, or else only one person could be there at one time. And so it was a struggle, and obviously that made it much more challenging to get work done because, and then the other thing that happened is I went on sabbatical. And so Rachel was very independent and motivated and took a lot of initiative to get things done. And she really did really successfully at San Francisco State. She got lots of grants, 8 grants and scholarships, and she was also a Genentech scholar. So she managed to find funding for herself the entire time, which was really, it's not so easy these days to be funded at San Francisco State, obviously. She also was really instrumental in getting another project off the ground, the one that you won't hear about today. She was working on avian pox in hummingbirds. So there was one other student, Aoife Galden, that was working on avian pox in hummingbirds, and she went to graduate school in Utah. And so we're like, oh no, we need to get these samples done. And so Rachel volunteered, and she did a great job extracting lots and lots of hummingbird feathers and testing them for avian pox with the PCR protocols that were established by Hannah, who you may also know. Right there she is, Hannah. So Hannah had established a PCR protocol in the lab to detect avian pox. And so you didn't know that you were using the protocol that Hannah had established in the lab when you were doing that. That you didn't know that you were going to be working in the same department. And so that was a really, a lot of work, and it ended up in a nice publication, which she's an author on. So obviously, most of my students end up doing a lot of work with birds. And this is, I think, one of the few trips that she did with birds. Here we were catching some birds right before she even started at San Francisco State. She was volunteering to come out with us and do some mist netting. And here you can see us all with masks. I don't know what happened to the masks in this picture. I think this was a little bit later. And we had a really great little picnic and camping trip, which was a lot of fun. So yes, Rachel didn't do as much lab work. I mean, she did much more lab work than she did in the field work because we basically ended up with samples that had already been collected at the International Bird Rescue. And so she got a lot of samples that actually Carlos was also helpful in collecting at the International Bird Rescue in Fairfield. And so Becky should be online, and thanks to Becky for getting all those samples and organizing for this project, which was started before Rachel got here. It was started to find out that these birds had these different parasites. And then we're like, oh, we can write another grant to find out what's happening when they have these parasites and when they get oiled and when things happen with these common murres. And so we'll hear a lot about that today from Rachel. But we got a grant from the OWCN, the Oil Wildlife Care Network. And so we got some money to do this project, and Rachel helped write that proposal, and we were funded. And so that's what started off this project. And it was a very clear-cut project. And of course, it was difficult. You always don't know what samples you're going to get, and it takes longer than you think, especially when the lab is closed down. And so I'm really grateful for Rachel for getting all this work done. In the meantime, she was doing a lot of other stuff. She also did a lot of work with herps, and I think she did some field work. This is not something that I know about. Here's this little lizard biting her. I don't know lizards at all, but I know that she was doing some exciting work in the Big Basin area, and then also in Oregon. Yeah, so she was doing some work up there. So she managed to capture a real research intensive program, plus also do some field work on the side with different programs. So she was really doing a lot of things. This is the lab that we had when she started out with Faith, Rachel, Aoife, and Roberto. And here's the latest lab with some new people, Carlos, Autumn, who are here, Caroline, and Eileen, and Ariana, and of course, Grant. So we're all pleased that we can have Rachel to carry on the knowledge, pass it from one generation of lab students to the next, which she's been so helpful in doing. And we're so grateful for Rachel. And now we're so pleased to see her getting a position here at the CalACAD, which will be, and she'll be a tremendous resource here for all of the participants of this meeting and at this lecture, and also the people who work at the CalACAD. So with that, I'm just pleased and honored to have worked with Rachel for these last 2.5 years or so. And I'm sure you'll enjoy hearing her talk. Welcome, Rachel.
Are we good doing? Good. Okay, cool. Okay, so thank you for that introduction. I want to start off by saying welcome to everyone here, everyone online. It means a lot. And I thought I would kick things off with a personal story. So back in 2013, I was a high school intern here at this museum. And I looked at the conservation biology in Xenopus longipes, which was an endangered African frog. This photo was taken on the opposite side of this wall. And now almost a decade later to the month, I'm here defending my thesis. So just a full circle moment and just an integral place in my science journey. So going through an agenda, we'll start with the introduction and the background information on this project, followed by the methodology. We'll do a deep dive into the results of what I was able to find, and then go into final takeaways and conclusions for the project. So starting with the introduction, fossil oil, also known as crude or petroleum oil, can have significant impacts on the environment. And spills occur for a variety of reasons, including transportation, offshore drilling, and industrial activities. For wildlife, this can lead to massive environmental damage, any contamination of food sources, and direct harm. Habitat destruction of aquatic species such as coral reefs and wetlands are extremely common and can cause long-lasting impacts on the ecosystem. And any inhalation of the toxic fumes can lead to respiratory problems and other health issues, which would result in breeding and migration disruption. Oil can coat the fur and feathers of animals, making it hard to maintain body temperature and buoyancy. And approximately 10 million gallons of oil are spilled each year. And while major spills can capture headline news, the vast majority are small and go unnoticed. So local organizations play a crucial role by responding and rescuing wildlife with the hopes of preserving and protecting them for years to come. One of those organizations is the International Bird Rescue, which was founded in 1971 by Alice Berkner after 800,000 gallons of crude oil spilled in the San Francisco Bay. They operate 2 centers, one in Fairfield, California, and the other in Los Angeles, and then a 3rd center on an as-needed basis in Anchorage, Alaska. Each wildlife center is made up of pools and tanks and has a full hospital clinic and run by a staff of veterinarians, nurses, and volunteers. They care for an average 4,500 wildlife annually. And birds are brought in for a variety of reasons, including starvation, anemia, trauma, and oil spills. Patients such as this pelican are treated for their symptoms and injuries. In this case, it would be oil contamination, and then cleaned and rehabilitated before their eventual release. And then fun fact, if you ever want a relaxing screensaver or a calm video, there are live streams into the pools of these birds. So you can just see them splashing around, playing with each other. It's similar to like the fire stream on Netflix. So, and for over 50 years of work, IBR has responded to over 225 oil spills and crises on every continent except for Antarctica. They're a member of the Oiled Wildlife Care Network, which was the organization that funded this project, and worked with a variety of birds, such as hummingbirds, hawks, owls, but they do specialize in treating seabirds. And I think there's this common misconception that Dawn dish soap is like the solution just saving all birds. Like you can see the cars, the commercials driving around. And while they do play a crucial role in that, it's organizations such as IBR that go out, send full staffs out to really do the work and protect and save these wildlife. So bird feathers are naturally waterproof, but in order to do so, each feather must be properly aligned. The barbs and barbules within each feather can hook together like Velcro and overlap, similarly to shingles on a roof. Not allowing water or air to penetrate through the feathers will ensure that the bird is buoyant and insulated from the cold. However, it's up to the individual bird to maintain its own structure. While preening, birds will keep their feathers supple by distributing natural oils, which helps maintain alignment and keeps long-term care possible. When oil sticks to a bird's feather, it'll cause the feathers to mat and separate, which can impair waterproofing and expose an animal's sensitive skin to extremes in temperatures, and that would result in hyperthermia or hypothermia. Instinctively, a bird would try to remove that oil by preening, which would result in the ingestion of the oil and cause severe damage to any internal organs. They will lose their buoyancy and often beach themselves in an attempt to escape cold weathers until aid and rehabilitation is possible. And long-term studies have shown that contamination can cause significant population decline, reduced breeding success, and delayed breeding times. Seabirds spend the majority of their lives out in the open ocean, so oil contamination poses a significant threat to their overall livelihood. And here are some of the photos of the common seabirds that the International Bird Rescue treats. You have albatross, egrets, herons, and penguins. The list and numbers of treated animals for both birds and other species, not just during oil spills, but in general rehabilitation, is constantly growing. And then we have common murres, our main focus for today, in the bottom right. So urea algae, or also known as common murres, are known as the flying penguins due to their tuxedo look. They're dapper black and white seabirds with slender wings that allow them to fly long distances to forage and swim to depths up to 100 feet. They're found in northern oceans and present all year long in the west coast, and then mainly in the winters on the east. Breeding on the Pacific coast ranges from the south of Alaska all the way to the Channel Islands in Southern California. And in the Bay Area, you can find them breeding along the Farallon Islands and the Point Reyes headlands. Like all seabirds, they remain primarily in the open ocean and rarely come to land unless they're nesting. And so this photo shows on the right a breeding adult, signified by the full brown and black coverage around the head, and then on the left are babies. Babies and non-breeding birds have a white stripe that either coats their throats, chins, and can wrap all the way behind their eye. In recent decades, North America populations have noticed declines, but Atlantic populations appear to be increasing. In addition to oil pollution, common murres are extremely vulnerable to gill net operations, overfishing of prey species, and the introduction to other mammalian predators such as foxes. And global climate change poses a risk to ocean temperatures and currents, which would have catastrophic effects, not just on murres, but on all seabirds. Common murres are regular patients at the International Bird Rescue for symptoms and injuries related to chronic oil contamination, anemia, and starvation. In 2015, several hundred common murres were brought in as part of a mass mortality event that were emaciated and had worn, frayed, or broken plumage, and they required prolonged stays in captivity to molt. In routine blood work taken from a subset of those animals, abnormalities were found with high total plasma protein values. Those slides were examined and hemoparasites were found and suspected to be either Babesia uriae or Plasmodium, which is a common avian malaria parasite. And then in 2019, a survey given by Wilmer Amaya Mija, which was a former graduate student of the lab, and Rebecca Duer, a veterinarian at IBR, they screened for Babesia infections. And Hemoproteus and Plasmodium were not found, but the number of Babesia infections were surprising. Of the 134 individuals sampled, 42, approximately 31% of that population, were positive for Babesia uriae, of which 4 were oil contaminated. There remains a large gap in published studies regarding immune suppression and general immunology in seabirds, and even more so, there's minimal information and research on their transcriptome responses. A transcriptome can be defined as the compilation of all the messenger RNA expressed in an organism to create and quantify complete sets of transcripts at a specific developmental stage or physiological condition. They'll vary depending on cell type, environmental condition, or stimulation, so it essentially provides a snapshot of genes at a given moment of time. Understanding of the transcriptome is essential for interpreting the functional elements of the genome, revealing the molecular makeup of cells and tissues, and understanding development and disease. One of the main approaches to studying a transcriptome is using RNA sequencing or next-generation sequencing. Assembling a transcriptome will build and reconstruct the full-length transcripts or sequences of RNA molecules. The goal is to accurately and efficiently assemble the full range of genes to be further analyzed for any changes in expression. Gene expression is known as the quantitative evaluation and comparison of those transcript levels. So any change in gene expression can be indications of health impairment. Upregulation, or an increase, and downregulation, which would be a decrease, refers to changes in gene expression levels in response to a particular stimulus or condition. The transcriptomic analysis provides the bird's eye view and the comprehensive report of all upregulated and downregulated genes. Depending on the stimuli, changes in expression can be short-term or long-term. So for the purpose of an example, on the left, you have a regular transcriptome response, and then on the right would be a response, say in this case, after infection. Each tick mark represents more mRNA found in the gene. And we can see that in genes A through C, there's an upregulation or an increase, and those genes may be involved in an immune response or inflammation. And then at the same time, genes D through F experience a downregulation, and those may be involved in metabolism and protein synthesis as a response to nutrient deprivation and stress. So the expression would be considered the collective change in all genes. Prior research has shown that the transcriptome responses will vary depending on an individual. When introduced to the same stimulation as a healthy individual, those with pre-existing conditions or some form of compromisation may respond differently. So a relatable example of this would be COVID-19. Healthy individuals may go through the regular 3 to 5 days of symptoms, while individuals who are immunocompromised, older, or with pre-existing conditions faced much more severe effects. And so here we have the same example of the previous slide, of a regular response, a healthy individual's response, and then we have a compromised individual's response. While the same genes exist, ones that originally were upregulated or downregulated switched directions. Studying an individual's transcriptome will show which genes are expressed in the response. So there have been a limited number of studies on the transcriptome response as a result of oil contamination. The first study looked at flounder in relation to crude oil spills with Vibrio anguillarum, which is a known fish pathogen. When analyzing the transcriptome response, there was a downregulation of genes related to immune function and hemostasis, including a reduced expression of immunoglobulin M, the main antibody found in fish. Results showed that immediate exposure prior to infection resulted in a 94.4% mortality rate, while those that were unexposed had less than 10% mortality. In another study, sea otters exposed to the contaminants of crude oil showed an elevation in genes directly associated with tumor formation and disease susceptibility. While there have been a few studies on the effects of oil contamination in wildlife, there are even fewer done in seabirds. The first transcriptomic study in seabirds was done on seaside sparrows when exposed to a petroleum oil. Researchers found an upregulation of genes relating to cell proliferation and liver regeneration, and a downregulation of genes relating to necrosis and apoptosis. In addition to oil contamination, a transcriptome response can also be affected by disease and infection. Babesia is part of a genus of protozoan parasites that infect the red blood cells of mammalian and avian hosts. Their primary vector are hard-bodied ticks. Babesiosis, the disease caused by Babesia, is typically transmitted through an infected tick bite, with symptoms including fever, fatigue, muscle aches, and anemia. In humans, it can commonly present itself with other tick-borne illnesses such as Lyme disease, and treated with a combination of anti-parasitic medication. Its life cycle, like all apicomplexans, go through an asexual reproduction in the blood and a sexual reproduction in the tick. Cells are characterized by ring and amoebid forms and fan-shaped or tetrad schizons. And these slides show infected mammalian blood cells in the upper corner. Prior research has looked at the host response within mice, humans, and cattle. Most common would be Babesia microti, which infects white-footed mice and small mammals, and Babesia divergens, that infects cattle. Historically, Babesia has had devastating impacts on the cattle industry and domestic animals. Similar to Babesia divergens, Babesia bovis can affect cattle and is considered to be the most virulent babesiosis. It's been known to wipe out and devastate massive populations of adult cattle, but surprisingly, young cattle seem to be unaffected. Adults that did survive due to their innate immune systems displayed chronic infection symptoms. In recent years, Babesia has been seen to infect birds, and currently there are 15 known avian Babesia species. However, little is known about their symptoms. Infected individuals are brought into rehabilitation centers and have been seen with lesions in their lungs. However, Babesia-related symptoms are not the reasons for their admittance, meaning that this parasite does not or may not cause significant enough damage to an individual. The vectors for avian Babesia are also presumed to be ticks, including Ixodes uriae, which is a common tick that has the geographic range that spans the northern hemisphere. It's been reported on at least 50 species of seabirds so far. The parasite can cause disease through repeated cycles of proliferation. Within each cycle, several rounds of DNA replication can produce multinucleated forms. That nucleus is surrounded by a membrane that can protect against any chemical or enzymatic treatment, and that makes the extraction process a lot harder and more difficult to isolate the DNA involved. Because of that, a comprehensive and completed Babesia-infected genome of birds is hard to find. On the other hand, infected genomes of mammalian hosts are extremely common. And so this photo shows avian Babesia-infected blood cells, and the gray dot in the center would be the nucleus that wasn't present in the previous slide. Prior to 2009, there were 3 known species reported in seabird hosts. The first was Babesia poliae, found in brown boobies, Babesia piercei, which was found in the African and little penguins, and Babesia benneti, found in the yellow-legged gull. Most recently, and the 4th Babesia identified in seabirds, and our main focus for today will be Babesia uriae, found in common murres. Given the potential for some seabirds to travel long distances, they can play an under-recognized role in the dispersal of ticks and other tick-borne pathogens. So there were 3 main objectives for this study. The first was to develop a full transcriptome assembly for Babesia-infected or oil-contaminated birds. Due to the nucleated red blood cells, a solid infected bird genome of Babesia does not exist. So we hope that these transcriptomes can serve as a stepping stone into acquiring genomes of more species of birds and provide a foundation into further genomic studies. The second was to compile a comprehensive analysis of the transcriptome response and the gene expression. Due to the virulence of Babesia uriae, we suspect that gene expression will be impacted as measured by the transcriptional response. By determining whether the prevalence or severity of Babesia is associated with oiling, we can compile a comprehensive analysis of RNA expression. And the third was to characterize the similarities and differences between the responses. We know that there will be differences between the 2 sets of assembled transcriptomes for each condition. And we hypothesize that there may even be some similarities. What those similarities are and the reasons why was something we intended to explore. All right, so moving on to the methodology. All samples used for DNA extraction were stored in a lysis buffer upon collection. Using Promega's DNA extraction kit, a total of 91 samples were extracted for DNA. and taken from a 2008 paper written by Breyer Bostrom from Texas A&M, PCR primers and protocols were used to screen for Babesia DNA. Samples were run in duplicate, with the first round producing 25 positives for Babesia and the second round producing 22 positives. Samples used for RNA extraction were stored in a trizol buffer, which essentially just preserves the RNA, and using QIAGEN's RNA-Z kit, a phenylchloroform extraction was performed to address the buffer. Total RNA was first screened on the qubit to check concentration levels, and then assessed on the bioanalyzer for quality control before being sent off to Novogene for library preparation and sequencing. Due to the cost of sequencing, it would not have been financially possible to sequence all the samples taken from IBR. The instability of RNA poses a significant challenge in any gene expression study, so oftentimes an RNA integrity number or a RIN value is used to ensure that the quality is high enough before sequencing. Each bioanalyzed sample generated a sheet of information as seen on the left, and the two main areas to focus on are RNA concentration and the RIN value. A baseline of 5.0 was first used to eliminate any samples that had deteriorated too much, and we found that selecting 3 individuals can provide a generally broad and accurate overview of a population. So we selected 3 samples with the highest integrity numbers for each of the 4 conditions for a total of 12 samples, as represented here on the table on the right. And it's important to note that sample 221341 was originally brought in for starvation. At the time of sample collection, the individual was noted for Babesia infection with either fish or vegetable oil present on its feathers. However, we recently found that the bird was misidentified and should be excluded from further analysis. So a total of 66 samples were extracted, with the 3 individuals having the highest RIN values selected as representatives for each of the 4 conditions. This table goes into further depth to show when they were collected, where they were found, and the reason for their admission. And then again, an asterisk has been used to denote the correction. Files were released and downloaded from Novogene and then uploaded onto the servers here at the California Academy of Sciences for storage and assembly. FastP was used as the FastQ data pre-processing tool for quality control, trimming of adapters, and generating reads. Then the 2 forward and reverse files were run through Spades to create the contig files, and through Quock, it was run to provide the meta-analysis. And then all programs were run on each of the 12 transcriptomes each of the 12 samples to generate the individual transcriptomes. So this table shows the meta-analysis for all those samples. And there's 3 main areas to focus on: total length, total contigs, and the predicted unique genes. Contigs refer to the set of DNA segments or sequences that overlap to provide a map that reconstructs the original DNA sequence of a particular region. So total contigs here ranged anywhere between 14,000 to 45,000. In addition, total length size ranged from 85 million to 174 million base pairs, meaning that these files were huge. So a potential future project may be to look at gene regulation, which is the process used to control the timing, location, and amount in which genes are expressed. And finally, when we inputted these files into Quock, the program generated its own prediction into the unique genes, ranging from 62,000 to 110,000. And so analysis of the transcriptome response was done with immense collaboration with Carlos Esperanza. Raw FASTQ files, along with a common mirror reference genome that was taken from NCBI, were uploaded onto the Use Galaxy bioinformatics platform. Trim-o-matic and FastQ was used to trim the pair-end reads and provide the quality control. And then we aligned the pair-end FastP files to the reference genome using HiSat2. Feature counts was used to assess the gene expression of each sample measured against the annotated gene. And finally, files were run through Lim of Room and DSeq to perform the 2 differential gene expression analyses, one to test the effects of a Babesia infection and the other to test the effects of oil contamination. So moving on to the results. From May 25th, 2021 to August 10th, 2022, 194 common murres were admitted to the rehabilitation center due to injury, starvation, or oil contamination. Of those, 193 birds were sampled and surveyed, 122 were considered to be after hatch year adults, and 123 were either euthanized upon arrival or within 24 hours of admittance. Comparing the 2019 study done in IBR, where they saw an average of 20 birds admitted per month, we saw a decrease of 13 birds per month. However, the euthanized rate increased from 50.7% to 63.4%, and the success rate of released individuals significantly decreased from 39.6% to 19.1%. Overall, the rate of oiled birds stayed relatively the same and remained lower than the prevalence for regular rehabilitation patients of non-oiled common murres. But we determined that oil contamination is correlated to the morbidity associated with Babesia infections in common murres. There were approximately 15,000 expressed genes in the entire common murre genome. There were a total of 2,121 differentially expressed genes with a statistically significant p-value of less than 0.05. 667 of those genes coded only to Babesia infections, and then 1,454 only to oil contamination. Roughly 57% of those were upregulated and 43% were downregulated. For p-values less than 0.01, there were 119 highly significant genes in Babesia infection and 239 in oil contamination. Of the significant genes, there were 1,724 with known information or characteristics as represented by the Venn diagram. About 1,000 of those were differentially expressed only to contamination, and about 400 only to infection. Between the responses to infection and contamination, 194 were found to be differentially expressed in both, which accounts for roughly 29% of the overall genes. And so this heat map depicts the top 100 differentially expressed genes by the adjusted p-value in birds infected with Babesia. The three left columns show the control birds, the three right show the Babesia infected samples, and the color of each gene indicates the level of change in expression. The darker the color, the more extreme the case. So a darker blue indicates a downregulation, and a darker red indicates an upregulation. On an initial glance, there aren't many particular trends. Similarly, this heat map depicts the top 100 differentially expressed genes, but in oil-contaminated and non-contaminated birds. Each row represents its own unique gene, and looking at this graph, the responses within the individual control and contaminated groups remain consistent to each other. When comparing the two heat maps, it's clear that having either a Babesia infection or being oil contaminated elicited some form of a transcriptome response as noticed by the change in color. While the Babesia heat map doesn't show a particular trend, the oiled heat map shows a significant trend in the upregulation of genes when a bird is contaminated with oil. It's also worth noting that the involved genes remain quite similar to each other. Now, this volcano plot shows the significant differentially expressed genes in Babesia infection. The x-axis represents the log of the twofold change in expression, and the y-axis represents the negative log of the significant values. Genes are represented by either a red or blue dot. The horizontal dashed line is the threshold of values where genes are considered significant, and the vertical dashed lines represent either a half or double fold in expression. So all genes differentially expressed above or outside these lines are considered to be of greater value. Genes of interest are labeled, including the genes that were differentially expressed in both tests. Similarly, this volcano plot shows the significant differentially expressed genes in contaminated and non-contaminated samples. Again, the more left or right, the greater the change in expression is considered to be, and the higher the lower, the greater the significance. Looking at the two plots side by side, the same genes are labeled in both, and it's clear that expression significance is slightly different. The labeled genes of interest all remain the same on each of the volcano plots, meaning that they were regulated in the same direction for both conditions. However, the differential gene expression in Babesia-infected birds had an overall higher significance and expression for fewer genes, as shown by the less condensed dots, whereas those that were oil-contaminated expressed more genes, but at a lower significance. Now, going back to the Venn diagram, there were a total of 1,724 significant genes with known characteristics. 194 of those were shared by both conditions, and of those 194, 6 were expressed in opposite directions. And these 6 are listed here with their descriptions. That means that 97% of the shared genes were expressed in the same way. Responses to specific stimuli would elicit a lot more variation in the activation or suppression of these genes. A much less likely scenario would be that all or most of the 194 shared genes were expressed in different directionality. So we hypothesize that this large amount of similarity may indicate that the function of these genes are involved in a generalized stress pathway, and that infection and contamination within birds may elicit similar transcriptome responses. All right, so moving on to the final takeaways. There were 3 main conclusions from this study. The first was that we successfully assembled the full transcriptomes for 12 individuals representing 4 different conditions between Babesia infection and oil contamination within common murres. The data set for this project can serve as a foundation for future studies, not just in common murres, but in any genomic study to other species. Next, the transcriptional responses for individuals was measured, and gene expression was seen to change in response to a condition. When analyzing each transcriptome response, impacted genes of high significance were identified, and the level of change in expression was measured. Finally, responses were compared and contrasted between conditions, and the expressed genes, including significant and shared ones, were identified. The shared genes between 2 conditions were noted, including the ones expressed in the same or opposite direction. And then one more note, it's not up here, but I just wanted to stress the novelty of this study, as it is the first time that anyone has looked at the transcriptome responses of Babesia-infected seabirds and then also comparing them to oil contamination within seabirds. And now for some next steps and projects on the horizon. So the first was to compare differential gene expression in birds, both parasite-infected and oil-contaminated. So again, we only looked at the gene expression for birds with either condition. So analysis from this project can be expanded upon by performing a differential gene expression test to determine how a combination of factors can affect a common murres response. We would use the Babesia-infected group as the control group and would test against the group with both parasite infection and oil contamination. Similarly to this study, we would determine which genes, if any, are turned on or off in response to oil contamination while already undergoing an immune response to Babesia infection. And we suspect that oil contamination and ingestion could potentially exacerbate the severity of the immune response by overproducing pro-inflammatory cytokines. Next is to determine the biological functions of differentially expressed genes. So gene set enrichment analysis, or GSEA, is a method used to focus on sets of genes to determine if they share commonality in regard to biological function. Data from differential gene expressions can be analyzed using GSEA and be tested for the overrepresentation of gene ontology terms, or GO terms. GO is a bioinformatics initiative whose goal is to produce controlled vocabulary that pertains to specific biological functions across eukaryotes. By determining if there is an overrepresentation of certain functions from entire groups of differentially expressed genes rather than individual genes, it can provide more insight into the biological pathways associated with a particular stimulus, which in this case would be infected or oiled samples. Having a stronger understanding of those functions would narrow down the use of phrases and terms within a particular stimuli. The third was to investigate the novel Matryoshka RNA virus in parasite-infected birds. Researchers recently discovered that the Matryoshka RNA virus appears to infect malaria-causing parasites, both in birds and in humans. Roberto Rodriguez was a former graduate student in the lab, and he identified the virus in association with avian hemosporidian parasites. Babesiosis and malaria are known to share common biological processes across organisms. So this gap presents an opportunity into understanding the molecular biology of these viruses a little better. And this can lead to a transcriptome response analysis between birds that are infected with just a singular parasite and birds that are co-infected with the parasite and the virus. And we did perform an initial screen on our 6 positive Babesia infected samples, and we did not detect the virus, but that doesn't mean that it might not be there with other species. And fourth is to publish our data and findings in a peer-reviewed journal such as Parasitology or Molecular Biology and Evolution, and that manuscript is currently in its works. So for some final notes, the consistent admittance of oil-contaminated and Babesia-infected common murres at the International Bird Rescue provides a long-lasting opportunity to study these birds and other seabirds in unique circumstances. Again, this served as the first time anyone has looked at the transcriptome response in these birds with these particular conditions, so we hope that this project will lay some of the groundwork for future genomic studies. And finally, by increasing our understanding of the health effects and internal responses of wildlife, we can better protect them and preserve them, not just them, all common murres as well, for years to come. And so there's a few people to acknowledge. First, the funding for this project was provided by the Oiled Wildlife Care Network. And then I want to give a huge thanks to Carlos Esperanza, who was monumental in the transcriptome response analyses, and Roberto Rodriguez at UC Davis for his help with the assembly processes, as well as current and former members of the lab, and then Rebecca Duer and the staff and volunteers at IBR, of which this study would not have been possible. They were the ones who collected the sample and the individual bird data, and just worked tirelessly every day to respond to oil spills and rehabilitate wildlife. And then to Jacob Gourneau and Jack Dumbacher here at the museum for their help with the bioinformatics work, along with the IZG, herpetology, and CCG departments here. And then my advisor, Ravinder Segal, and committee members, Rebecca and Jaime Chavez, for their advice and guidance and expertise throughout this process. And then finally to friends and family who have supported me throughout the last couple of years, listened to me talk, to give me advice, go for coffee chats, not just like in the Bay Area, but the East Coast and even in Singapore. And I know there's a couple of people watching. And then finally to the digital engagement team for making this in-person thesis defense possible. And so with that, I will take any questions.
So a lot of these samples.
Oh,
sorry, thank you. Hi, Rachel.
Hi. It is.
Hi, Rachel.
Hi.
Thank you. So most of these samples seem to be from birds that were after their hatch year, correct? So can you talk a little bit about how the response was in the contaminated chicks, if there were any?
So we had one contaminated chick in our subset of 12 samples. It was for a non-oiled Babesia positive case, I believe. Like an elder individual, a transcriptome response is also different in comparison to younger chicks, and they're harder to find. That isn't necessarily to indicate that they don't survive, but they may be more severely impacted. So finding individuals that are both Babesia positive and oiled will probably be a lot harder. But I would assume that their transcriptional response would be much different than any adult that we looked at.
Thank you for your talk, Rachel. It's great. So Babesia and the oiled condition are like two seemingly like kind of extreme circumstances that these birds could be in. Do you anticipate that there are any other sort of conditions that you would see another like transcriptomic gene expression response to? Like, I don't know, I'm just thinking about like wildfire or other sort of, any other extenuating circumstances that you would expect some sort of response from these birds to.
I would assume that like any stressor, even if they're injured, like a broken wing or something, would provide an additional stressor. So, and that would have impacted the transcriptional response. So anything that you could think of, not just wildfires, but if they're co-infected, injured, starved, or anemic, all of those would be factors that would play a role in the response.
Thank you.
To kind of piggyback off of that, would you expect a similar DD results from other stressors, or do you suspect that the DD results you found relating to Babesia and oil contamination to be specific to these types of stressors?
So I only did a super general screen for the genes that we were identified. The most common was protein coding. So I'm not totally sure, but I would assume that if it was an immune response, that those would share similar responses in genes elevated. And then if it was like an environmental factor, I feel like they would be different, but not entirely sure. I did not do the deep dive into the genes just because there were so many.
Hey Rachel, thanks for a great talk. So I had a question about, it seems like your research is like a really cool intersection of like working with organisms and vectors that interact with people, as well as helping you understand like the ecology of like a natural system as well. What are your most exciting takeaways for you personally, for both the kind of more human related as well as the more natural ecology related?
I think in trying to understand like any pathogen model organism, the hope is to provide like a piece to that puzzle. So babesiosis that can infect humans, and then even looking at Ixodes uriae, which is a tick found commonly in other species, just providing that snapshot of how they affect common murres has been really, really cool. And then Babesia infects just so many other seabirds in and of itself. So being able to now say that I could compare that to other species is really cool.
Joining remotely, Eric asks if there were any differences in age or size of the birds in different treatment groups. In the sample collection? Yes.
Okay. I would say the majority were adults. I did not actually have the experience to collect them. That was all done by IBR. But I think that the majority were adults in this size. I don't know if that answered the question. If that's Professor Routman, hello.
Great talk, demonstrations. I just wanted to hear from you, when you have two comparisons of gene expression, it is clear when you have your oil-contaminated birds that the response is almost identical across your three individuals. But for the babesiosis, there seems to be some intra-individual variation.
Yeah.
What do you make out of it? Do you think some individuals have a less, how do they, this variation can be explained versus the two treatments that you have?
So with PCR, it just detects like whether an individual has the Babesia DNA present or not. I think that the level of infection may vary, not just within an individual, but its age, how long it's been infected. And so that may be an indication as to some of the discrepancies of the different lighter, darker colors that we saw. In comparison, I think the oil contamination can be easily or more clearly defined as a yes or no is it present? So that's one of my hypotheses as to why there were some discrepancies in that.
We have a few more questions from people joining remotely. Christina asks if there are any similarities in transcriptome responses in relation to other Babesia species.
I didn't have the full chance to look at that, but I do know that, for example, if we were to look at the Babesia transcriptome responses in penguins, those are flightless seabirds, so that may have a completely different impact than common murres that can travel longer distances. And then I know that in cattle, there were a different set of upregulated genes, but I, again, didn't compare the genes that were identified between the two.
And Sid asks, besides reducing oil spills, what do you imagine preventative work to look like in the future based on your research?
That's a good question. I don't know. I think the hope is that there are greater case studies that can provide a greater idea into what oil spills and oil contamination does, and then working with either agencies or organizations to kind of provide a voice to wildlife that may not otherwise have it. I think just through the process of this study and working at the museum, the ultimate goal is to protect and preserve organisms and wildlife. And so I hope that this can provide even just the International Bird Rescue with the larger case study when they're doing their outreach and work.
There's no more question. I want to thank again Rachel for a beautiful talk. And afterwards, we're going to sit, Jaime, I, and Becky online will talk about a little bit about, how we were going to evaluate this. But I think there's going to be a party afterwards, right? Somewhere or no?
Everyone is invited to Underdog Trace. I don't know if it'll be a party,
Not a party,
but
just
it's
fun.
Cinco de Mayo. So.
Cinco de Mayo. So we're going to go to the taco place?
Yeah.
Okay, we're going to go to the taco place afterwards and meet there probably 4:30?
4:30-ish, yeah.
4:30. So thanks again, everybody, for showing up on the Cinco de Mayo on a Friday afternoon, and really congratulate Rachel again.